Welcome everyone to We Science. With Science, our goal is to promote local science on a global scale. Today, we have a special guest, Associate Professor Arutha Kulasinghe. Arutha is a global leader in across many different fields, especially personalized medicine, as well as spatial transcriptomics in cancer, as well as in COVID. And Arutha is a close friend of mine, and we have met for maybe 10 years. And I met him when he was at the TRI, Translational Research Institute. He was doing his PhD, and then in less than a decade, he's now a global leader. So thank you, Arutha, for accepting my invitation to be part of this podcast.
No, thank you, Carlos. It's an absolute pleasure. I'm super excited by the invitation and being able to be a part of this forum with you. And we go a long way back. So I'm looking forward to the conversations.
Oh, fantastic. Well, I'm very excited about it. Now you are a leader of the clinical omics lab at the Fraser Institute at UQ, University of Queensland. So I just wanted to start. What motivated you to become a scientist? I mean, you are originally from South Africa.
Yeah, so I grew up in all parts of Africa, Carlos. So, Dad worked for the UN and a few other places. And so we traveled all the time through Africa. And one of the biggest killers in Africa is HIV, malaria, and so on, right? So a lot of people die and are impacted by infectious disease. But I was shaped really early on in my career. I was about 10 years old, and my grandfather had colorectal cancer, and he was in Sri Lanka. So we went there, and they were performing the surgery at 2 a.m. Mom and I were sitting outside, and the surgeon came outside with this silver stainless steel bowl and showed us the football-sized tumor that my grandfather had. And so I remember I had this watch with a camera on it, and I took a photo of that, and it just remained with me. And then I was involved in his aftercare, and you can't sit and you can't eat and all these challenges. And then till he passed, and so I realized really quickly that I wanted to understand translational cancer research, develop better diagnostics, therapeutics, and so on. And so part of that journey was to actually leave Africa to come to Australia to really understand cancer diagnostics and therapeutics. And so I was that was sort of a moment that shaped me really early on, but it's been the foundation of, a lot of people asked you, what are your whys in life? And it was one of those things. So when we go to work nowadays, it's not a formal role that we do it for the, to actually move the science forward and hopefully into the clinic one day.
That's a great story, Rutha. Yeah, particularly when we are cancer scientists, that we wanted to make an impact, right? And sometimes we forget there's patients there that are needed. And a patient could be a father, is a grandfather, is a brother, sister. So it's very personal.
Yeah, so Carlos, I remember shortly after, also during my PhD, I went and spent some time in the US and, visiting labs and stuff like that. And in one of the labs, and you'd recall Peter Kuhn, who worked on circulating tumor cells and still does, he had patients actually doing research in the lab, right? So they were centrifuging samples, they were looking at blood assays. And I spent a lot of time with the lab there and really understanding how patient-centric they are in their research. And you're not sitting on Facebook or Instagram or anything when they come into work. They're actually working because they are intrinsically motivated by having patients as part of their research. And so it's something I've been trying to implement back home in Australia, obviously. How can we increase patient advocacy and patients as part of our research? So all the patients we work with, I tell them, whenever you come for a checkup, please come into the lab, knock down doors, ask students what they're working on, ask postdocs what they're working on. And that really motivates the lab more than anything else that we can sort of formally, put out.
I think when a cancer scientist or any scientist see a patient is life-changing, because it makes it to have a purpose, to be able to find a cure for, not particular for the particular patient that we are seeing, but also for the future patients that are coming. And
Yes.
your presentations, you always mention about it takes a village. I really like the expression. And then it cannot be more patient-centric than that, inviting patients to run the research, to do, to be involved.
Yeah. So when we get bloods, Carlos, they're always warm when we bring them into the lab because they've just been collected from the hospitals. And you'd know that, right, with a liquid biopsy background. And so I tell the lab, this is liquid gold that we have. these are precious patient samples that we need to extract the maximum information from. And so I always tell my students and mentees that, this is liquid gold that we're working with. And I think they get it. I think they so hopefully some of them find that interesting and want to pursue this.
That's great. They're part of joining the army against cancer.
The village.
So what was your academic journey? So your passion is cancer, across other things. So how was your academic journey from bachelor to PhD?
Yeah, so I did my undergrad in medical microbiology in South Africa at the University of Pretoria. And then shortly afterwards, I did my honors there too. And the honors was on sexually transmitted infections in third trimester pregnancies and the effect on newborns and so on. And so it was really interesting because I learned a lot of different skill sets, learned to interact with pregnant moms and their families, and so that sort of shaped some of the experimental work that we were doing. But I really wanted to get into sort of cancer biology, try to understand it at a greater depth. And so that's when I moved and applied to Australia to do my PhD. And so I did that at the Queensland University of Technology, where I met you, Carlos. And I was working on essentially a liquid biopsy. And liquid biopsies are looking at measurements in the blood, circulating tumor cells, circulating tumor DNA. Today extracellular vesicles and sort of, that sort of space is really hot. And so we were describing what is found in the circulation by using different types of microfluidics, different types of enrichment in the blood, and then what was the prognostic implication for that patient. And so some of the work we did was initially in lung cancer and head and neck, looking at circulating tumor cells in the blood. And we found that, these aggregates or clusters of tumor cells tend to associate with a more aggressive phenotype. So patients that do really poorly have these clusters, and we think that's a protective function of not just tumor cells, but also having immune cells, so it can protect sort of recognition from the body's immune system whilst it's transitioning from your primary cancer to your lungs, liver, brain, bone, and so on. That's what I did my PhD in. And what I really learned, and I'm sure you can echo, is that liquid biopsy is super interesting, but it's super rare, right? These cells are extremely rare in the blood. And so part of that thinking was, I need to go back to the tumor microenvironment. I need to go back to the tumor. Every time I'm looking at these cells, I'm like, where did these cells come from? How are they, breaking off the primary cancer? Which part of the tumor is it breaking off on? And so these are sort of unanswered questions that gave me sleepless nights for many years. And so shortly after my PhD, I realized that I need to go into the tumor microenvironment and try to understand the primary cancer better. At the same time, a lot of these new tissue imaging approaches were just hitting the market, right? So you could look at 4 or 5 colors on tissue. And so, when you think of immunohistochemistry, you look at 1 or 2 stains, right? And so now all of a sudden you can look at 10 or 20 proteins directly on the tissue. So that's what I thought would be, the next wave to really understand and characterize the microenvironment. And so that's what I built the lab on, took the risk that it's a brand new space, brand new field. But I thought that information that we're looking at, it's not, we're not grinding tissues anymore. We're not, dissociating tissues anymore. We're seeing the tissue directly in the space in which it's found in the patient, right? When you go to buy a car in a dealership, right? If you were to see all the parts laid out, that's not a car, right? But you actually want to see that car in its entirety. And so that's what this sort of field has done: spatial, transcriptomics, proteomics, interactomics. And so that's what I built my lab on. It was just me and my postdoc, James Monkman, and that's when COVID hit. So yeah, we pretty much loved them.
You were mentioning about liquid biopsy, because the premise of liquid biopsy is be able to because the issue is that there are different tumors in the patient, right? And they are different, or they tend to be different because of the intrinsic diversity of those tumors. So if you biopsy one side, even if you take a biopsy from one portion of the tumor, it might be different if you go 1 millimeter to the right or to the left. So then the beauty at the time, when there was all these big, announcements in terms of achievements and liquid biopsy, was to be able to, in the blood, be able to get all the information from the different tumors, basically like a pool, like a soup, coming from the circulating tumor cells as well as the circulating tumor DNA, the exosomes, and et cetera, that provides a non-invasive. But as you said, Arutha, I agree that the issue is dilution. Unless we are able to concentrate those, and we were able to, when we were working on microfluidic devices, we were able to enrich those circulating tumor cells and be able to profile them in collaboration with Majid. But that was a fantastic initiative and also work. Understand? But yeah, there are some challenges in everything that we do, right?
Yeah, no, completely agree. I used to, obviously your career, I used to look up to a lot of the work that you were doing in the microfluidic space, and no, it's fascinating, right? I mean, I think we need better tools, and a lot of the microfluidics helped us to capture those cells and to characterize them. But I just feel that it's such a diluted, at least the cells, right? And I think the tumor DNA that's used clinically today, EVs are super popular at the moment. But yeah, part of my challenge was when you look at these cells down a glass slide, yeah, where are they coming from in the tissue? How are they getting there? You were seeing the shift in EMT and all these processes, stemness and all of that. But yeah, my dream experiment is to be able to see the primary, the met, and the blood all together. But we have to start somewhere.
Yeah, and you mentioned head and neck cancers as well as non-small lung cancer.
Yeah.
Could you please expand on briefly
Yeah.
on to a person who doesn't know much about those cancers?
Yeah, so head and neck cancer is made of multiple subtypes of the head and neck region. So you have the oral cavity, back of the throat, sort of oropharynx, larynx, and so on. So they call those regional, that grouping of tumors, head and neck cancers. And so head and neck cancers have been on the rise. They are getting better with therapies, traditional chemoradiation and immunotherapies are starting to become more first line in clinical trials now. It's mostly given in the metastatic setting and recurrent setting, but now it's starting to come more first line where you have a better, I guess, shot on goal to get response in these patients. One reason we are seeing an increase in head and neck cancers is a lot of sort of HPV driven head and neck cancers are on the rise. This is due to oral sex and things like that. And so the tonsils are these sort of crypts that can have virus residing in those regions. And it was an area of research that was very active in Brisbane at the time. And so that was the reason we got into it. Obviously head and neck cancers, you don't hear about it regularly, through your traditional media, but these patients can't eat, they can't swallow. Often they have 10, 15 centimeter tumors taken out, and they can't speak, and so it's a very visual disease too, and so it's an area that we wanted to focus on, in addition to lung cancer, to really be able to characterize the different microenvironments of the oropharynx, the oral cavity, tongue, the lip, and so on, and really understand the different processes that are happening, but also being able to describe them in detail. And we also see a lot of cutaneous skin cancers can metastasize to the head and neck regions. Getting that information is really important.
Fantastic. So then when you were working on the PhD, so then you were very well set to be able to become like a global leader in the liquid biopsy, and you already explained why you moved. And you got this prestigious fellowship by the Australian government. What factors, thinking about just the PhD, what factor do you think that contributed to your success as a PhD student and set you in the right path to be able to succeed forward?
Yeah, that's a really good question, Carlos. I think we often in academia, by the time we realize what we need for the opportunity, it's too late, right? When you're applying for a fellowship or something, you realize, oh, I actually needed a number of first author publications. By the time you're applying for a PhD scholarship, there's certain requirements that you need to have met. So what I saw from day one of my PhD was, we didn't have a lot of funding for my work. And so part of the challenge for me was to, I was grant writing from week one. I was trying to collaborate from week one, seeing all these really interesting technologies come to the market, but then also being able to work with industry, collaborate with industry, leverage some of the clinical cohorts that we had collected locally, and being able to really test on system with some of these technologies. So one of the things I did quite successfully was we worked across probably 8 to 10 liquid biopsy, circulating tumor cell enrichment technologies at the time to push through lung, head and neck samples and different types of enrichment. And so in so doing, we were able to not just benchmark, but contrast and compare what are we actually capturing from the blood. And so I worked really closely with industry, worked really closely with patient advocates, and worked really closely with clinicians to be able to characterize in small cohort studies, almost cross-sectional studies, what was in the circulation, what were the circulating tumor cells in these populations in a cross-sectional study. So, we were able to publish about 8 to 10 publications on the topic during my PhD. And so that really set me up to be able to apply for a fellowship. In addition to that, I was working, very closely with industry and clinicians to be able to really nurture those relationships. I remember one clinician I worked with, I actually wrote a grant because I wanted to do a project on lung cancer because I realized that there are a lot of circulating tumor cells in small cell and non-small cell lung cancer. And so, wrote the grant, took it to the clinician, and the clinician was running this really big lab, super successful. And he called me and he said, in my 30 years of research, not a student or a postdoc has ever done that in collab, been able to, write a grant end to end and share it with me like that. And so that's where that collaboration started. And so we went back and forth, refined it, and finally got some funding to get it off the ground. And so that's when the lung cancer work really took off. And so I think, being able to maintain, strong collaborations with clinicians, I think clinician scientists are, the super rare breed, but I think being able to nurture, as a translational scientist, the links between the work you do on the bench and the clinical work that they do and finding solutions for their unmet need, right? So what are the challenges they have? And then trying to figure out, is there a way that you can develop an assay that can potentially help, a challenge they have clinically, right? Why do PD-L1 low tumors sometimes respond to therapy versus PD-L1 high? What is it in the blood that might be predictive of therapeutic benefit or outcome? And so working with these sort of clinicians really nurtured my growth. And so that was sort of the next step. And so by the time the fellowships, by the time I finished my PhD, I had sufficient publications to be able to apply for a fellowship. I initially wanted to go to the US, go do 2 years, visit Minyu or Peter Kuhn's lab and spend some time with them. But I think serendipitously almost, because that would have been very close to COVID, I went and visited those labs, but was able to apply for the Peter Doherty NHMRC ECF and was successful, straight out of my PhD for that.
Fantastic. Wow. So then you sort of move into transcriptomics, spatial transcriptomics. You mentioned initially about that, and then COVID. Please tell us why. You already mentioned that you wanted to investigate, go back to the tumor biopsies, and be able to understand better, to identify biomarkers that we could use in the clinic as a prediction of responses. And I just wanted to know, how was the transition?
Yeah, it was quite steep, Carlos. I think during the end of the first year of my postdoc, I realized that's when I was working really actively with a number of clinicians in this space. I realized that what we're seeing in the blood is a very small snapshot of what is probably coming from the tumor. And so being able to do multiplex analysis on tissue might be a solution to understand the microenvironment better, right? One of the biggest challenges still then and still today was, are there better predictive biomarkers than PD-L1? Because all they knew was CD8 infiltration into the tumor was predictive, but not much more information, right? The two sort of biomarkers are PD-L1 expression and CD8 infiltration and maybe total mutational load. And so what I wanted to do was go into the tumor, map every cell by cell. And so the technologies were also just hitting the market. And so I applied for this internal grant. It was a small $10,000 grant. I remember I could run 3 samples because they were about $2,500 each, and it gave us, I think, about 24 proteins worth of data of a single section. And so I went back, got the data, went back into the analysis, and we were looking at the, you can see the co-expression of these genes and proteins vary across different spaces in the tissue. And that was an aha moment. It was like, this is why we need to map every cell and get that information, because you can imagine, right, if a tumor in this corner metastasized or this corner metastasized, you need to know where that's coming from in space, and having that information is going to be useful for therapeutics, for understanding what's driving metastases and so on. And so really low dimensional data, but on 3 samples. And so That was the first study in head and neck cancer, and James, my postdoc, was working on a small project in lung cancer, really looking at the data QC, data normalization, developing a number of these QC metrics for spatial data. And then COVID hit. And so we were two of us, and we thought, what do we do to, what do we do during COVID? And I've always tried to be an agile person, Carlos. I've always tried to think sort of outside the box. I think we need, as early career researchers, we need to be agile. We need to be able to pivot. I was working with a very close collaborator from Brazil who was actually based at our institute, Fernando Guimarães. He's an associate professor now. He's an NK cell expert, and I met him at a Cure Cancer event, and he said, we've got these tissues from autopsy studies in Brazil. They can only do single-plex IHC, maybe dual-plex IHC. They don't have the resources, but they've got a lot of samples. And he was arranging MTAs and things like that in the background. And I said, hold on, Fernando, what if we were running spatial on this to figure out what is the impact on different organs with COVID, right? Mm-hmm. And so the idea was, localized virus, you're using RNA-scope, and then look at the transcriptomic and proteomic changes cell by cell in these tissues. And so they sent us lungs, hearts, placentas, and then working with Johns Hopkins, we were able to look at brains. And so the brain study is still in prep, but we've published across other studies. We were able to localize where is the virus in the lungs by mapping cell by cell. And then we were able to identify the changes happening locally in areas of infection versus areas that are not infected. So we're looking at the sort of local changes in the tissues. And then the beauty of the study was we had multiple pandemics as well. So we had the 2009 flu. And so we can contrast COVID to flu, the 2009 flu. And so, in pregnancies, third trimester pregnancies, we could see preeclampsia was up. In cardiac tissues, we could see DNA damage was up in COVID-19 patients, which was in stark contrast to flu. Flu was eliciting this sort of inflammatory response, but COVID was eliciting this sort of DNA damage response, which is very similar to cancer, right? And diabetes. And yeah. Wow. And so for us, it was so much fun because we'd get the data, we're looking at the lungs, we're looking at the virus localization, and now I'm calling pathologists, I'm calling virologists, immunologists, and all of a sudden we have a call of about 20 people, intensive care specialists, looking at the tissue in real time on a Zoom call or Teams call or whatever it was, and then trying to tease apart what's happening. And so we replicated that model. And so I think indirectly we built these really well-functioning teams of people that like to work together on different projects. And so, I'm not a virologist by training, nor an immunologist, but I know some of the best that can contribute to this work. And so the infectious disease work is ongoing at the moment, but it allowed us to, get some funding for the team. It allowed us to expand our network. It allowed us to collaborate with the US, Brazil, and then there were, on the back end of a lot of these studies, we had clinical diagnostics teams from Sydney and so on that saw some of our tissue-based findings and came to us and said, hold on, we've seen these changes in the blood. Your data is showing that this actually came from that part of the tissue. And so one of those changes was this IFI27, which is a type 1 interferon. We found this in comparing the lungs of COVID patients to that of normal flu, 2009 flu, and normal controls. We found this one gene was constantly elevated in patients that did poorly. We then went into the blood and then pre-Omicron screened about 1,000 patients from 8 countries for that single gene. And we could see that IFI27 measured within the first 3 days of symptom onset was a predictor for how bad you're going to be, whether you need hospitalization, whether you need ICU, and so on. And so working with a company in Sydney called SpeedX, they've actually developed a finger prick test for this. I think that shows you, why it's important to, not I hate the word pivot, but collaborate, but also contribute to global challenges. And so we're very open-minded when it comes to research. We love working on different ideas, but we need to bring the domain experts. So my lab during that time, Carlos, became a computational lab. Because of the data volumes that were coming through, we realized, okay, the wet lab, we go in, we generate the data, but now we spend 2 years analyzing this data.
And that's a fantastic example. It's a great story. And it's a fantastic example about seizing the opportunity. The pandemic affects all of us.
Yeah.
But seeing the opportunity when we were fighting to get into the buildings,
Yeah.
you adapt quickly, then be able to sort of build from that and be able to find the right collaborator, the right samples, the right time. We
Yeah.
discussed this for the last time that we met. Is about riding the right wave, properly, and be able to at the right time with the right people. So it's fantastic, and
Yeah.
it's a great story about what we could do as a scientist to be able to take advantage of these opportunities.
I think it comes back to the village, right? We need to build that village around us of people, like-minded people that can get together around a problem. I recall, we had these cardiac samples come in, and I have no background in cardiology, right? But I knew that John Fraser was leading one of the total artificial heart transplant works in Brisbane. And so I pinged John and I said, look, John, we've got this, really rare cohort of cardiac tissues from autopsy studies. And, John's incredible. He was able to get us some funding within a couple of days to actually do the experiment. Had we put this through, your traditional funding mechanisms, it would have taken a year or two. And, then the pandemic's probably moved on, but we could actually get in the lab, order reagents straight away within about 48 hours to actually do the work. And so those are the opportunities that open themselves up. And John and I recently, collaborated together to build the Queensland Spatial Biology Center. And that was on the back of that cardiac COVID work. Just two clinician scientists and a scientist just coming together around the problem and trying to figure out, using cool science, what can we discover about this disease?
That was a fantastic story. Thank you for sharing it, Arutha. So
Thank
I
you.
just wanted to know if you could please share in lay terms why is the spatial transcriptomics and your current research, because I know that that example about the COVID during COVID is just the beginning.
Yeah.
And doing a fantastic stuff, work and research. You're driving science, to the ages.
Yeah.
And that's fantastic, Arutha.
Yeah, so I mean, for the spatial transcriptomics, Carlos, that's sort of, the oncology is our focus, right? Head and neck and lung coming through the PhD was our main focus. And it was really about how do we identify the changes that are happening in the microenvironment cell by cell, but also identifying patterns at the RNA, protein, H&E level, integrate those data sets together at a cohort level to identify predictors of therapeutic benefit to immunotherapy. The challenge is the same, right? So immunotherapy for solid cancers is one of the biggest challenges. You don't know which patients will respond, which patients won't respond. It's the same for ADCs, right? Antibody drug conjugates that are, currently being used in therapy. There are no predictive biomarkers, so the change remains the same. The challenge is, how do you describe the microenvironment that drives those patterns of response and non-response at a sort of prognostic level, but also when you look at the transcriptomic data, you might identify mechanisms driving resistance or response, right? So, are there additional drugs that might be useful in those patients and so on? So that's been the major focus of the lab over the last, 2 or 3 years. We have a really strong focus on transcriptomics, whole transcriptome, and more recently, single cell and subcellular resolution. These are, call us 10-day runtimes per slide, about 30 terabytes of data per slide. And so you can imagine, analyzing and data storage, compute, and all those challenges. But it's getting really interesting because we're identifying potential mechanisms driving resistance to immunotherapy. We've identified in early stage, in advanced stage lung cancer, we've identified this one pathway called ferroptosis, which is a iron-dependent cell death pathway that is being, your tumor cells are almost adapting to the stresses of IO, and then they're turning on different pathways to enable their survival. But we can see that these are low infiltrated tumors immunologically. They have this sort of resistance pathway turned on. And so what we're trying to figure out is, these are metabolic processes that are happening in these tissues that are resistant to therapy. And so what we're trying to figure out is, are there combinations of metabolic inhibitors in combination that will improve therapeutic benefit to those patients that respond? So immunotherapies typically work in 15 to 30% of patients. Identifying predictive biomarkers for those patients is important. But what about the 70% that don't respond? What are the other pathways that are targetable using, repurposed drugs or anything else that's out there? And, metabolic inhibitors, Carlos, it's not anything fancy. Metformin is a metabolic inhibitor, right? So we're seeing that these tumor cells are adapting to the environment. And so what we're trying to figure out is how are they adapting? What are the pathways that are being turned off and turned on? And can we target that's, highly complex data sets, at the RNA level, at the protein level, but we embed that data onto an H&E. So we have ground truth, we have a pathologist's interpretation of the tissue, we have pixel level resolution on the omic integration onto the H&E. And a lot of the work that we're trying to develop is, can we build foundation models around this to really be able to leverage the power of spatial omics, to be able to scale that. Remember, we can run these experiments in very privileged labs around the world, but in Africa and a lot of the sort of third world, they can't run these expensive experiments. So part of my motivation is growing up in Africa is everyone's can do a 50-cent H&E stain. Any lab can do that around the world. They can take a photo on an iPhone, and there are tools now being developed with collaborators at Harvard and so on, where you can pretty much talk to that image and ask it, what am I looking at? It'll say this is lung cancer. It'll say this is the mutational profile. It'll say this is, adenosquamous, just based on an LLM overlay on that H&E once you've trained it. So now you've got the pathology chat model, and all you needed was an H&E stain, which costs 50 cents. Any lab in the world can do that, and people have got a camera that can take a photo of that slide. So the idea is to democratize this, and I hope that we will go that way one day. But obviously, we need to run the discovery work up front to be able to inform down to a pixel level on an H&E what that looks like. I think H&E gets the spatial work into the clinic, right, when we can draw on that. But that's a really cool part. That's what I'm excited about.
And then you also work on pandemics.
Yeah, so this is on the back of the COVID work, Carlos. So we again, good people. This is Kirsty Short, Larissa Labs, and others, Gabriel Bells, and so on, where they realized the work. These virologists came to us and they were like, we've never looked at tissue before. We, traditionally we'd look at swabs and blood and so on, but we've never really been able to look at the tissues. And so we had a student, Lauren, who went and spent a year in Germany and Austria in museums digging up FFPE archival collections of Spanish flu tissues, right? And then she brought these tissues back, and so we had multiple pandemics, about 5 pandemics over the course of about 100 years. And the question was, adult versus children's flu affects the children and adults differently across the different pandemics. And so the question was, can we map the lungs across each pandemic and then contrast it over time to see how it differentially impacted kids and adults? And so we ran this crazy experiment on a 105-year-old tissue. We had the block. The block, Carlos, was just black, just pure black. We sectioned it. We had a fresh internal cut. We ran the whole transcriptome and about 600 proteins, pseudo-bulked. And we were able to get beautiful protein data. We were able to get about 21 colors on multiplex data. It made the cover of the Australian, they called it Science is Cool, which was really nice. So we can get RNA and protein data of 105-year-old tissue, the block's intact. We had the pathologist annotation for each of the regions that we profiled, and those regions separated out transcriptomically on the PCA. So the histology was actually mapping to the transcriptome data. So that was real. So that's showing that it's not, it's the signal we're actually getting is real. The work is still being put together, but that was really exciting because it's imagine what this means for, hospitals and archives and pathology labs. You don't need to go and collect prospectively. You can go to your clinicians, your pathologists, and say that, look, we have looked at tissues that are 100 years old. Can we go back 50 years or 20 years or 30 years and look at certain cohorts that they have just got banked in their biobanks, right? So it really unlocks the power of biobanks and retrospective cohorts. Maybe there was a drug they tested in the 80s that worked in a subset of patients. We can go pull those, right? Whether it's melanoma or another disease, we can pull those. We know the clinical data. We have the tissues, and now we can actually robustly say that we can get the RNA and protein off those tissues. So I'm excited by that. I think that, we no longer need to go and, spend years and years and years collecting tissue and then collecting all the follow-up data. We can actually go back 5-10 years, pull different cohorts, different drugs that might have worked, didn't work, but also, potentially working with pharma too, right? It really unlocks samples, clinical data. So yeah, that was the purpose of the work, but it's ongoing. Again, Carlos, I think one of the things that I realized sometimes when you're looking at the data in these lab meetings, I have to take a step back and I'm like, what are we looking at? Is this infectious disease? Is this cancer? Because, Carlos, the same pathways are coming up, the same genes are coming up. And so
Wow.
we think about these in different, we think about, this is head and neck cancer, this is flu, this is lung cancer, as separate baskets. I think, the changes, at least immunologically, are very similar. And so sometimes the things we're learning can be informed from spaces that we're not directly looking at. One thing I can say without taking too much away from a paper that the lab is putting together is that we looked at a recent study of lung cancer patients given immunotherapy in the first line setting. We could look at every cell type, NK cells, CD8s, macrophages, cell by cell in the lungs, and contrast between responders and non-responders. The typical, pathways came up, the typical, inflammation, B cells, lymphoid aggregates, all of that came up. One of the really interesting bits, without giving away the story, was there was a whole finding in infection. So there is a role of infection that actually plays a role in certain cell types actually being there in the lungs. And so this was fascinating work. And so now we can draw on all the infection experts that we have. And so this is where it gets fun, right? This is where science becomes super cool. I'm not a domain expert. I need to work with a lot of people and a lot of people that are a lot smarter than me. But I think it's such an interesting space where you can leverage these domain experts to tease apart the disease that you're looking at from a single lens, but now you've got this multi-lens breakdown of the disease. So that paper will hopefully come out end of September, and that'll be a fascinating finding into why some of these cells might actually be in the lungs in the first place.
That's very interesting, Arutha. I'm very fan of your research. Awesome, and also because you were mentioning before that COVID was different. That was, DNA damage compared to the flu. So it will be interesting to see across the different pandemics to be able to see and also prepare for the future pandemics, right?
Exactly. I think the question is, what can we learn from existing diseases that prepares us for the future, right? And so our goal, Carlos, was never to become infectious disease researchers, but it was to contribute to a global pandemic, right? And I think, it enables so many collaborations around the world, around Australia too, and now we can actually connect the dots. So yeah, and it's funny, whenever I'm on calls with the virologists, they keep saying that, oh, you're coming over to the infection side. But now when we're looking at the cancer data, we're actually seeing that we actually need them for the interpretation of this. So I think it's this really fluid space where all our domain experts sort of, we have to learn a bit of computational biology, biostats, informatics, immunology, virology. And I think, the days of you saying that you're this type of scientist are gone, because you need team science. You need that village. A bioinformatician, Carlos, can't do it by themselves. I certainly can't do it by myself. But you need everyone to come together to be able to understand what you're looking at.
Fantastic. That's awesome, man. So science, as you mentioned, right, this is changing more now than ever. Initially, there was before scientists, we were working and people might be thinking, listening to you, oh, why spatial transcriptomics, all these multi-omics, etc. But basically, initially, the scientists, we were just working on single genes. And Arutha mentioned about doing a single staining. When you take it at the biopsy, they were just checking couple of markers individually, but then in genome-wide association studies, macroarray gene expression, SNP arrays, then becomes all the age of sequencing with RNA sequencing, genome sequencing, whole exome sequencing, and then after that, everything was done on the tissue. So basically, as Arutha was mentioning before, all the tissue was disrupted and then be able to dissociate it. So we didn't have the resolution at a single cell level. Then all these single cells, which it happened in the last decade, single cell, we were able to profile cells at a single cell level and be able to, instead of having this bulk resolution, we had that identify immune cells within the tumor, etc. And then we were just digesting the single cell. I was working on implementing CySeq, single cell proteomic as well as RNA. And then Spatial came. Basically, it's taking a picture. And when you see these pictures, one of the pictures from Arutha is this tumor that has 4 different parts, and ones that are more infiltrated, the others have like a different prognostic signatures from each one of them, which was a great, is a beautiful image, and be able to see what's happening. Whenever you dissociate a tumor, you lose the geography of those cells, who they are interacting with, who they are nearby, and how that makes the pathogenesis. So then it becomes, and we applying all these omics strategies, and you're doing, transcriptomic, proteomic, genomic, epigenomic, etc., interactomic for biomarker and drug target discovery. But what do you think will be the rationale for this in terms of applying all these different layers of data?
Yeah, no, great question, Carlos. I think at one sort of level, right, it never has been sufficient. So with the technology advancements, we've needed to stay up to date. But the challenge is looking at the different omic layers individually doesn't seem to give us the complete picture, right? So it needs to go back to this multi-omic approach of the tissue and potentially the blood. And so how you would do that is, you'd look at your transcriptomic data, your proteomic data, or and your H&E, and then being able to integrate that data is really important. And now, there are methods to do, epigenomic mapping and so on. So one of the challenges, Carlos, is, obviously with single cell work, your endpoint is a UMAP or a TISNI, right? If you see a lot of CD8s in an area, you might say this tumor is highly infiltrated, right? That's your endpoint.
Yeah.
On the tissue, if those CD8s are on the other side of fibrosis, they're not infiltrating, right? So this is why the tissue mapping is fundamental, right? We need to, every cell you call on a TISNI or UMAP space needs to be able to be projected onto the tissue, and you have a pathologist interpretation of that to say that that is a CD8, that is an NK cell, or whatever it is, right? So you're responsible. You can't get away with, this is my UMAP, that's my cell type, right? You have to be able to say confidently and work with a pathologist to be able to interpret that. So that's where I think the power of Spatial is. But also, it's not just the multi-omic data sets, right? Now you've got this multi-omic data set overlay on the tissue, so you have this omic cloud. But also, some of the work the lab's been doing recently, which is just super exciting, is using some of the AI models on this, right? So now you can feed in simple binary inputs, right? Relapse or no relapse, for example, right? So in that data set, you can use sort of contrastive learning models to now plot on the tissue where the areas of relapse are likely to be high or low, right? Using this sort of wealth of spatial data. And so I'm like, we think quite linearly, right, around these problems, but I think bringing the AI and into this, and some of the machine learning pipelines and the deep learning pipelines that are being developed, there are going to be leaps and bounds in how we can overlay the clinical data into this. A recent project we worked on, Carlos, was just a proteomic overlay on spatial proteomics above H&E. And one of the inputs for the data set was relapse, no relapse. But at the protein level, we had metabolic, functional, and immune cell types, right? And tumor cell types. I gave this to one of my really talented computational biologists that is PhD on the CERN Super Collider, so single molecule imaging. And he came back and he showed me the cell type map, right? So we know where the tumor cells are, the B cells, the T cells, and all that. And then he plotted another map. And this is early stage resectable lung cancer, right? Which is curative by surgery alone. But then we had 3-year follow-up as to whether the patients had relapse or no relapse. He then had a second row of images, and this was relapse risk plotted on the tissue, but relapse was a binary input into the model, right? So this is now showing you areas in the tissue which are likely to be high relapse or low relapse, but your input was a on-off, right? And so we went back, I brought in, a bunch of pathologists, a bunch of biostats and computational people in the team. And I said, Aaron, explain this logic. It can't be, is it circular? And so we went through it, and now we started to project known biology, right? So what is this learning? So we put on B cells and TLS-like markers, and we could see, okay, it's lighting up in the areas associated with low relapse. This makes sense, right? So this is where it gets interesting, is where it's not just the depth of the information, it's the inferences we can make with clinical information. So I think this is such an untapped space. This is where the foundation models and all of that's heading, right? So if you take a model and you take 20 scans of H&Es for skin cancer, and you scan another 20 scans of spatial proteomics data, if you put a blank unstained slide in there, it should be able to call proteins and all of that off that unstained tissue. And so I think a lot of this modeling and AI work is going to get super, super exciting going forward. And the things, I love bringing in people with different backgrounds. So Aaron's from a physics background.
Yeah.
It's like, there's no biology there, which is brilliant, because now it's like there's no sort of bias in his interpretation. So these are the sorts of things I think we can sort of learn from going forward, but they'll also make really large knowledge gains in how we're interpreting this data.
Yeah, that's fantastic. And particularly like complexity, right? The tumor is very complex, and you need to be able to dissect that complexity. You need these tools. One of the problems that I was thinking, and I wanted to ask your perspective on this, is that to make a discovery, groundbreaking discovery, you need to apply all those different tools, and they are not cheap. Certainly, they are not cheap. And my concern is, my question to you is, it feels like it's going to create like a gap, because of course the expectation is more with the money, right? But also it may create an imbalance in the research groups, because access to the technology due to funding, because that is always get me the more information with a cheaper price. And what is your thoughts about that?
Yeah, I mean, if we go back, Carlos, I started my lab with $10,000 and 3 samples, right? And then leveraging that data, the collaborations, that then snowballed, right? So I think you need to start somewhere. And the beauty of this field is that a single lab cannot do it by themselves. And so you need multiple different teams coming together to be able to understand it. So some labs might have a wealth of, tissues. Some labs might have a wealth of, immunology expertise. Some labs might have computational strengths. And so, if you can contribute, like we did for the COVID work, towards a common goal of we're trying to understand this disease cell by cell, then everyone comes together. So I think, obviously, living in Australia, our teams are quite small, right? And compared to the armies in the US, that they have really large, teams, we often think, how can we compete with those labs? But I think in our individual spaces, there might be super unique questions that we can answer based on cohorts and collections that we have. So I think you need to start somewhere. I fully understand what you're saying in terms of, scaling and the challenges there. I think for your disease that you're really passionate about, you should start, even if it's a 6-color multiplex IF stain, if you can understand your tissues and layer that data, because the beauty is we can strip and reprobe those same samples using multiple different assays. Sometimes we might not have funding for certain assays, Carlos, and then you might reach out to a collaborator and they're happy to collaborate with you to run those samples. Now all of a sudden you've got a data set where you didn't have the funding, but now a collaborator has provided you that data, and then you might be able to contribute people from your team for the analysis. So I think there are, these are ongoing conversations. None of us can do it by ourselves, but I think together we can. And so we need to share the love around and build these teams and work with each other collaboratively. I always tell, I always tell my team when we collaborate that, there's a win for everyone. There's a win for the computational person. There's a win for the immunologist. There's a win for the biostats person. There's a win for the PhD student. There's a win for the, each contributing member of this team. It might not be within the next couple of months, but it will be over the next 2 or 3 years, because you might get your main paper out and then there might be sub-studies that each person can do a deep dive on.
Yeah. So then you were talking about personalized and precision medicine, right? Some people prefer personalized, other people prefer precision, which means that it's the right treatment for the right person at the right time. Why is that important? Could you please share your perspective on that?
Yeah. So, I mean, when you look at certain therapies that are given now, Carlos, they're not individualized to the certain patient. The individual, like at a trial level, they've shown benefit, right? But the challenge is cancer is so complex that using these spatial mapping approaches, we use this term heterogeneity, right, Carlos? We use the word heterogeneity when we can't explain what we're finding, because it's so different. The beauty of spatial is that we can see heterogeneity. We can see there's this beautiful head and neck example that you were speaking about earlier. The sample almost has a north and south pole. The north pole has, tertiary lymphoid aggregates, CD8 infiltration, M1 macrophages, the sort of cell death pathways are all turned on there. But at the bottom, it's metabolically active, lacks tertiary lymphoid aggregates, has this M2-like macrophage phenotype, and is sort of the invasive or aggressive area of the tumor. So in this one head and neck tumor, you've got a north and south pole, and also the subtumors within it. So, where is that bit of the tumor that spreads, that metastasizes? So I think with cancer, every patient is unique, every patient is individual. And so being able to learn a lot more information about that individual patient and the trajectory over time, over the course of therapy is really important. The time is important because you want to give the right drug at the right time for that patient, right? When the patient is optimal to receive that therapy. It's all about trying to figure out in the patient journey, based on their unique tumor profile, what are the drugs that do you give chemo up front, or do you prime the immune system with the chemo before IO, or do you give IO up front? Often head and neck patients have had chemo, radiation, surgery, and then you give immunotherapy at the end, and often the immune system is fried, right? It can't work because you're expecting it to work really late, but maybe that needs to be dialed back. So that's the time component of it. One of the studies we're doing for skin cancer is, head and neck and lung, you can single time point biopsy. You don't really get serial time points. Skin is a great organ because you can biopsy the same site over the course of therapy and look at the microenvironment changes under the stresses of therapy. So that's some work that's ongoing in the lab. Skin cancer is obviously huge in Australia. So yeah, I think we're trying to understand each patient better to be able to identify predictive biomarkers for those unique patients.
What do you think are the current bottlenecks for the translation of those discoveries into a personalized medicine approach?
Yeah. So I think the adoption is really challenging, right? So we often work, I try to, rather than showing the pathologist the end results of the oncologist, I try to bring them on the journey. This is what we're looking at, this is what we find, this is the troubleshoot, this is what we're troubleshooting. I think in so doing, they learn what we're learning as well, because fundamentally, your predictive biomarker for relapse or response cannot be a spatial signature. It needs to be something that a pathologist can stain on tissue. For example, it might be positive in the tumor for 4 markers. So now you're actually just looking for 4 markers that are positive in the tumor, as opposed to a spatial transcriptomics or sort of high dimensional assay. So we really need to simplify these assays, develop the signature, build a minimum viable product or minimal signature that a pathologist can screen on to, identify patients for high risk of relapse or response to therapy, or patients that won't respond to therapies. Really, starting off in that inverted funnel approach and getting really narrow, and then being able to validate prospectively and then move some of that forward. And I think insurers and so on have a big role to play in this too.
And it's great, for example, what you share about taking a picture from the H&E and be able to sort of infer the different populations and then be able to find markers there. And also having the blood test, for COVID, differentiating how you're going to respond, etc. I think I'm very passionate as same as you with it has to be low cost. Be able to bring all that complex and also all the different high level, high resolution, ultra high resolution tools to be able to bring into something that could be cheaply implemented into the clinics, not just for Australia or the Western world, but also for countries that are in developing.
Yep, yeah, completely agree.
And we could talk here. I'm just skipping some of the questions because it's so fascinating. I could ask you a lot of questions about each one of the stuff that you're saying, but in the interest of time, I just wanted to keep jumping to the next questions. And then I just wanted to know, as a scientist, we have many failures and eureka moments. Could you please share some of the significant ones? And actually, one before you answer, when we met last time, and you were telling me this story about the different markers that you were using. That was one of the motivations to start this podcast, because our stories like that should be known by the general public, because people need to know that we scientists, we are always thinking of how we can make a bigger impact. So yeah, please share.
Thank you for that, Carlos. I think for us, when we initially ran the It was a low-plex, 7-plex, I think, 7-color vector panel on these tissues. All I was trying to figure out is, can I light up the CD8s and the tumor cells and maybe get some readout on Ki67 simultaneously? So we did all the opal staining of the tissues and took it into the imager and scanned it, and then turned on all the markers. And I had the H&E with me, right? So I'm looking at the H&E and I'm looking at the slide, and I'm like, wow, like you can see every cell lit up across that tissue. Every marker you can measure the intensity level of, where that is in space on the tissue. And so that was really my eureka moment as to why I needed to do this. It was sort of like, we have so limited information from the H&E. How do we sort of enhance that? And I think spatial proteomics enables that. So we started off with a 7-plex, and so that was the initial study, but now we're over 103 proteins on the same tissue section. And so you can imagine, the level of information we can now glean from these tissues. And it's not, for a pathologist to do 103 stains would be 103 cuts. You're not looking at the same cell if you're looking at 103 because you're moving in space. So we can get all that information from one sample. And recently we partnered with Abcam, and we've been able to look at 1,300 proteins, Carlos. That's all of Abcam's IO inventory. It's compartment specific, so it's not single cell, but we're now approaching mass spec types of readout using translational tools where this turnaround is antibodies by sequencing. The turnaround is about 8 days. So we're super excited. We published the 580 plex study that was in Tan et al. NPJ Precision Oncology, and now we're writing up the 1,300 plex study. So I think that's how it scales, right? In a translational manner. Abcam pretty much put most of their content onto it, which was incredible. But it's antibodies by sequencing. So we're super excited by how this is now a translational tool, and that's really scaling.
Yeah, I remember you told me that you had a box. It needs to work. It must work. And that's a fantastic
That's
story.
It. Yeah, that's it.
You were like,
yeah.
Nothing else but that, a box, a kit to be able to run it. Yeah. And on the other side, you always advocate for junior and emerging scientists, and you are very well, not just focus on the science, driving the science, but also be able to sort of build your team up, not just your team, but also your colleagues, your friends in the science, etc. And what motivates you for it? I remember that when I shared a post, and then the first person that I received a call was from you. I was like, Carlos, are you okay? And then, it's great to see not just a fantastic scientist, a pioneer, and also like a person that is driving research and science ahead, but also having this fantastic human being, a person that is able to sort of, we are humans, we all have issues and etc. And it's great to have people like you that is always building the future of the scientists in Australia. So what motivates you for that? And also, because that would be the last question is, what pieces of advice do you give to a junior scientist?
Yeah. So I think our journey is really tough, right, Carlos? It's a really, it's failure to failure to failure to small win to more failure. And so it's this constant hamster wheel that we're on, and we don't celebrate wins anymore. And I think it's something we need to. When I saw your post, I mentioned earlier that I used to obviously look up to you and all the work that you were doing, incredible work in Western Australia. And so part of it was we need people from diverse backgrounds and really sort of different ways of thinking about disease to be able to work together, right? And I think I always try to nurture EMCRs that are coming through, because I think they have the crazy ideas, right? They have the unfundable crazy ideas that no one will put a dollar to, but I love just listening to what would be cool in your world. What is the experiment we could do, and being able to lean on that and actually being able to support that as part of our academic journey, right? And part of being mentors. I think we need to do that. So for me, it's about crazy ideas, funding the crazy ideas, because I think incremental gain does not cure us of disease. We need people from diverse backgrounds, physics, biochemistry, astrophysics, in medicine to make the breakthroughs, right? That's where the really different types of thinking can work. And then creating space where there is no silly question. They can all ask their different questions and then figure it out. So part of that is trying to figure out. I love having lay conversations with anyone, just whether it's a taxi driver or someone that's just helping you, and they think outside the box, right? As scientists, we're trained to think within the box. They've always got really creative ideas, and then I come back to the lab and I'm like, what about that, what about this? I was in a cab going somewhere, and I heard about this. So I think that's where we can lean on new and innovative ideas, and that's where the breakthroughs really happen. The second part, I think, Carlos, is we need to be constant learners. I think none of us have the answers, especially in Africa, where when I grew up, education and training is often, it's not widely accessible, and so we need to constantly learn and have a learner mentality, because that'll help us get far in life, I think, and it'll also not just help but the world in all of the global diseases that we work on. I think having that learner mentality. When I go into a meeting and it's on infectious disease, I shut up and I listen to the domain experts talk about it, and then you absorb, right? You absorb and you learn about it, and you learn that flu is not just one pandemic, it's multiple. And I think that's super exciting. It keeps us refreshed, it keeps us motivated, and I think in the near future, hopefully there'll be better treatments for individual patients and multiple diseases. So I think having a learner mentality is fundamental to everything we do in life.
Yeah, and I had to ask this question because I remember when we were sharing the same field in liquid biopsy, we were talking, and then we had all this bunch of crazy ideas. We both are blue skies, right? And I also like the crazy ideas because I think sometimes we are constrained. I had a situation where I would apply for a grant, and then they told me that, oh, it's too blue sky. And then I planned the next year, and then no, it's too grounded. So then, how you, as a blue sky person who has been successful, how you bring those ideas to life, be able to bring that blue sky and bring it there. Because I think in funding, there is always this commercial, the idea of thinking, I say in the wrong word, but it's basically, sometimes we are just what could be possible between what we have. But if we wanted to go beyond, for example, you are unleashing your creativity with those pandemics, etc., and the spatial transcriptomics, etc. As a blue sky person, what will be your advice to bring those ideas to the earth?
Yeah, so I think you have to be creative in how you find funding for those mechanisms. During COVID, there were things called fast grants. These were grants that you could write a one-pager on, and within an hour you'd know whether it was funded or not, right? Because there was a need for that. I think we need to be agile and nimble in the different types of funding that we can apply for. Traditionally, drug dogma will tell you that you should apply for Cat 1 funding, sort of NIH and HMRC, DoD, and so on. But I think working with philanthropy and other organizations where they can take on that risk for a crazy idea, potentially angel investors and so on, where the risk to reward might be a higher risk model, there you might actually have some wins. So I think figuring out who that audience is and then working with them to fund your crazy idea, or at least give you the foundation to ask the question. And so you need to have a diversity in the funding mechanisms.
So yeah, thank you, Arutha. It's been so inspiring listening to you and all these different stories. And I'm very fan of your science. Always very pleased to see your success. And I know that this is just the beginning for you. And I know that the more will come. And I'm looking forward to see everything. And thank you for the generosity. I know it's a little bit over time, but thank you, man, for taking that time to share and be able to give us a little bit of a journey into your brain. And yeah, I'm very excited to see the future.
Thank you, Carlos. Yeah, no, thank you. And they say you should always give flowers to people that you admire. So thank you for all that you do. I think this is going to be an incredible journey for you.